Okay, welcome everyone to the EvoFunPath seminar for March. Today we've got as our speaker one of my colleagues at the University of Manitoba. So I'll just briefly introduce Dr. Georg Hausner. Dr. Hausner obtained their PhD at the University of Manitoba in botany in 1993, working on the taxonomy of the Ophiostomatales fungi, then pursued postdoctoral studies at Michigan State University, working on mitochondrial genetics in Neurospora crassa, the famous model fungus. Worked briefly for Agriculture and Agri-Food Canada and had a short stay at the University of Calgary before returning to the University of Manitoba Department of Microbiology to lead his own research program in the year 2000, focusing on the characterization of mitochondrial genomes for members of the Ophiostomatales, with an emphasis on the molecular evolution and biology of group 1 and group 2 introns. So very glad to have you presenting for us, Dr. Hausner, and I will turn it over to you.
All right, well, thank you very much for the invitation. And what I would like to do is probably more give you a bit of a flavor of the various things we're doing in the lab. And it's all about, in part, about mitochondrial genomes. But the focus is the Ophiostomatales, although I will sneak in another fungus which shows or highlights some of the work we're doing. Now, before I get there, and I just have to rearrange my screen a bit, I would like to respectfully acknowledge that I live and work in the original lands of the Anishinabek, Ininivuk, Inisinivuk, Dakota Yate, the Dene, and the Inuit, and the national homeland of the Reti River Métis. Now, Ophiostomatales, why? Obviously, they're an important group of fungi. Some members are causal agents of plant and human diseases. A lot of us are familiar with Dutch elm disease. However, some members of the genus Sporothrix are also potentially pathogenic fungi on mammals, including humans. Now, the taxonomy itself of the Ophiostomatales is quite complex. It's undergone extensive revisions over the last 40 years. And this is in part due to the extensive convergent evolution that has happened in these fungi and unrelated fungi where morphological characters evolved in adaptation to similar lifestyles. And I'll sneak it in now, because these fungi can be invasive, they can be moved around the planet, so to speak. Knowing what you have in your part of the world is important so you can distinguish it from parts or from fungi that have been introduced. Now, many of these species are associates and vectored by insects, such as bark beetles and other arthropods, and many of them are associated with causing blue stain on hard and soft woods. And that has problems when you want to sell the wood because exporting stained wood is sometimes problematic. Just a few slides of what these organisms look like, and I have to admit I would call this the Hollywood version of a member of the Ophiostomatales. They're usually characterized when they do produce perithecia or ascocarps. They're long-necked, sticky, slimy spores at the tip, and this is an important adaptation for insect dispersal. Here's just an example of a dead Dutch elm tree, North American elm tree, killed off by Ophiostoma novaulmi, which is a real problem, obviously, in North America. And even when they produce conidial state or asexual states, they usually are on long stalks with slimy, sticky spores. And again, it's an adaptation for insect dispersal. Quick little snapshot. Again, I talked about blue staining that can be caused by these fungi. And although it does not impact the structural integrity of the wood, it does sometimes reduce the value because it does limit export. Other countries don't want to import wood which shows signs of biological activity. Here's an example of a bark beetle gallery, as you would find under the bark of trees infested with beetles. And these fungi usually live along these bark beetle galleries and spend a good part of their life there, although some of these fungi also can spend part of their life cycle on the insect itself. Not going to dwell on it too long, but I do want to highlight the idea about the taxonomic history of these fungi. When I started a long time ago, there was one genus which accommodated everything. Then it was split into two genera, Ophiostoma and Ceratocystis in the 70s. And now we recognized, of course, in the 80s and 90s that actually these are totally unrelated. They belong to separate orders. And now, in the recent years, Ophiostoma has been subdivided into up to 19 different genera, with still some undefined lineages, so more genera to come. And Ceratocystis, on the other side, again, has been subdivided into many groups. So the taxonomy of this group is very active. Now, I'm going to focus mostly on mitochondrial genomes. Why are mitochondrial genomes interesting in fungi? Well, first of all, what do they look like? Well, mitochondrial genomes are obviously small and compact, as you would expect. However, they can be as small as 12,000 nucleotides. And a recent paper showed that they sometimes can be over 1 million nucleotides. So size variation is tremendous. They are usually depicted as circular molecules. However, it's been recognized they may also be linear concatenars as to how they're actually found within the mitochondria. However, there are truly linear versions found in some fungi, which even have telomeric-like ends. Each mitochondria will have several copies of this genome, can be 20 to 100, it varies. The gene content, ironically, across fungi for mitochondrial DNA is quite consistent. There's only about 14 core genes, plus or minus, dealing with, obviously, cytochrome oxidase, electron transport chain, NAD dehydrogenase subunits, ATP production, and the components needed for translation. Transfer RNAs, ribosomal RNAs, small subunit, large subunit, and sometimes a ribosomal protein. Additional features can be plasmids, introns of various types, and sometimes introns can encode proteins such as endonucleases and reverse transcriptases. I have to talk a little bit about introns because obviously that's what my lab does a lot of work on. The type of introns you find in organella genomes like mitochondria are usually referred to as self-splicing introns. So they're kind of, at the RNA level, they can self-splice. Now they do benefit from sometimes nuclear factors to promote the splicing, but they can splice on their own usually in a test tube, so they're kind of true ribozymes, catalytic RNA. We have group 1 introns and group 1 and group 2 introns, what we find in the fungi. They're differentiated based on their structure at the RNA level, how they fold, and they're differentiated based on their splicing mechanism. Now, very briefly, here's a group 1 intron drawn in a two-dimensional way, showing the upstream exon in a black box, the downstream exon here in a black box, and the intron itself at the RNA level will arrange itself in what we call paired P regions. Typically, we see up to 10 paired regions, and this is established by hydrogen bonding, so there has to be base pair complementarity, and the folding is essential. If they fold correctly, it brings together all the components that have to be next to each other for self-splicing to occur, which in group 1 introns is initiated by an external GTP. I do point out this P10 interaction, which is crucial as part of aligning all the splice junctions, which allow the intron to be removed and the exons to be spliced together. So P10, remember P10. Another feature about group 1 introns, and it also applies to group 2 introns, these are actually mobile elements. We call them mobile introns. They can be located inside genes, but they have mechanisms usually catalyzed by the proteins they encode, which allows them to move to new locations. Although usually it's site-specific, so these don't randomly move around. They usually go from one allele that has an intron to a cognate allele, which is an intron minus version. They go home. That's why we use the term homing a lot. So homing endonucleases are DNA cutting enzymes which allow basically a group 1 intron to move from one location to a new location. But it is still a homologous insertion site. It's very specific. Now, we use the terminology donor and recipient. So you may have an allele that carries an intron, so it's my donor. The intron encodes a homing endonuclease protein, which has to be, we have to transcribe it, we have to translate the protein, the intron can splice out, so that happens at the RNA level. But what happens at the DNA level is that this homing endonuclease is a site-specific DNA cutting enzyme. It'll cut an allele that's intron minus, generates a staggered cut usually. The staggered cut will be expanded by exonuclease activity, and this will initiate basically a recombination process for repairing this. So basically we involve the double-stranded DNA repair mechanism, and that involves homologous recombination, whereby the donor becomes a template for repairing the recipient. So what you're doing is you're copying the intron sequence into the donor by repairing it, and the intron now has found a new home. Group 2 introns, again, very briefly, they also form structures. They're best visualized as a 6-fingered hand, and the fingers are called domain 1, 2, 3, 4, 5, 6. Again, it has to fold into a proper configuration, and that will bring the flanking exons together and also the splice junctions, which are needed for the intron to splice out and the exons to be spliced together. So this is sort of an idealized diagram of a group 2 intron. Here's the upstream exon, and then in the white box here is the downstream exon, and in between the intron folded. Domain 1 is important, and again, that's one area to remember. There are sequences present in domain 1. We call them exon binding sequences, EBS. And the exon binding sequences, as the name suggests, will bind to sequences in the up and downstream exons, which will have, the exons will have corresponding intron binding sequences. At the RNA level, it allows the group 2 intron to assume a configuration, bringing together all the parts needed for splicing, and also it stabilizes its structure. Group 2 introns can encode proteins, reverse transcriptases, and these reverse transcriptases are important in, again, catalyzing the mobility. And again, this is site-specific. So again, we have an intron plus allele, and the intron will move to an intron minus allele. But here it's a little bit different. This has an RNA intermediate, so now we call it retrohoming. So what happens is, again, we have a gene with an intron, we get transcription. The reverse transcription or the reverse transcriptase open reading frame can be translated. We get this enzyme, which has several functions. The reverse transcriptase will bind to its own intron RNA, which assumes a lariat-type configuration. The two travel together, find a suitable insertion site. The 3' end of the intron will actually nick the target site, and the reverse transcriptase will nick the bottom strand. So we open it up, we generate a cut. This allows for the intron RNA to be inserted into the cut region, and it allows the reverse transcriptase to make a cDNA copy. And that's what's shown at the bottom here. And eventually the host repair system has to get rid of the RNA component, and everything is patched up. So basically it's an element that moves using an RNA intermediate. So all right, so we have mitochondrial genomes, we have genes, and genes can have introns, and these introns can move around. So what do? Well, we work on the Ophiostomatales, and we are interested in mitochondrial genomes, in part to look for these introns. In general, we have sequenced quite a few of them, obviously. So to summarize, the gene content, conserved. Gene arrangement, highly conserved, nothing special really. Size can be variable, anywhere from 21 to more than 150,000 nucleotides. That's what we've seen so far. Why the size differences? It's usually due to introns. So some have a lot of introns, some have very few introns. Some have what's called complex introns, and I'll talk about this a bit further. So why are mitochondrial genomes interesting? Well, why are introns interesting? Well, sometimes these introns can form interesting combinations. Because these introns are mobile and they move from an intron minus to an intron, well, from an intron plus to an intron minus allele, well, sometimes introns by accident probably insert into each other. So we have a group 1 intron inserting itself into another group 1 intron, or a group 2 intron inserting itself into another group 1 intron, and all possible combinations. And we call these novel. We call them twintrons. Sometimes people use the word nested introns. It's probably best to just call them complex introns because they're composed of several modules. And that brings me to one example. Obviously, I can show a lot. So I want to talk a bit about Ophiostoma ips, which was the work of one of my graduate students who actually is here today, Jigisha. Now, this particular fungus, Ophiostoma ips, it's a classic blue stain. It's quite commonly found in the northern hemisphere. It's usually isolated from conifers, vectored by insects. Not considered a primary pathogen, but it does have economic impact because of its blue staining activity, which can reduce the value of timber. Now, we sequenced the mitochondrial genomes of several strains and isolates of this fungus from North America, including a couple of strains from New Zealand. In general, they all have a similar structure. They're circular molecules. We're looking at a size of 110,000 nucleotides. So what's interesting about Ophiostoma ips, well, it's quite rich in introns. If you look at this diagram, it was generated by a program called Circus. The outer circle shows the genes and how they're arranged. And below it, you see these little blue boxes. For example, COX1, we have a lot of little blue boxes here. And all these blue boxes are introns. So you get very quickly the idea is that there's a lot of introns present in this mitochondrial genome. And actually, the intron make up a significant part of the size of this mitochondrial genome. As I said before, in terms of gene synteny content, very typical with regards to other Ophiostomatales. Going across the members of the Ophiostoma ips, we usually do what we make intron landscapes. And I have others, which would include all the Ophiostomatales, and it gets very busy. But I'm showing you this, that when you actually map the location of introns, you kind of get an idea that some genes are intron rich, like COX1 here. The size of the bar corresponds to the frequency of how many times we find the intron amongst members in this group of fungi. So this one is only found a few times. This one is found a lot of times if it's a high bar. Yellow are undefined introns. Red are group 2 introns. So some genes are intron rich, some not so much. So I'm not going to go into this too much, but there is a bit of a biased distribution of introns when we go across the fungi. What I want to talk about right away is 2 introns we found in Ophiostoma ips, which we find more interesting. There are these examples of where we have what we call nested or complex introns. So I'm going to introduce 2 examples. One is found in the COX3 gene, and the other intron configuration is found in the COB gene. Now in COX3, the second intron is something what we refer to sometimes as a tandem intron. So what do we have here? So first of all, COX3, there's an intron, and when you look at it very carefully, it's around 3,000 nucleotides in size, but it's actually composed of 2 modules. The first module in itself is a group 1 intron. Group 1 introns can be categorized into different families based on features of their secondary structure. So this is a group 1A intron, and it's followed by another module that also fits a group 1A type category. Each module includes a homing endonuclease gene, And homoendonuclease genes are named after conserved amino acid motifs. So this is referred to as a largely dark member, and this is a largely dark homoendonuclease. So what happens here? Now I'm going to make the story short because this one is not as exciting as the next one. Turns out that based on RNA-seq analysis, so we did transcriptomics here, we did take a closer look at the RNA of this particular bug, and we did find that in this fungus, this whole thing is spliced out as one unit. And this was not our terminology, they're named zombies. So when you have examples of twintrons, trintrons, multiple introns arranged together, but they're spliced as one, people refer to them as zombies because the individual modules have lost their ability to splice on their own. They have become a composite element. And so now they're spliced as a unit. Now we found another intron in this fungus, which we found quite interesting. And this one is in carb, the fourth intron in Ophiostoma ips. Now this one is a bit more complicated. Again, we have a group 1 intron module. Then we have a second group 1 intron module following it. The first group 1 intron module has a group 2 intron inserted. So we basically have 3 intron modules. Group 1 intron module, group 2 intron module, a group 2 embedded in the first one. 3 parts. So the question is, how does that splice? How does that work? So again, just to blow it up a little bit, I call this building complexity. These introns, when they start inserting into each other, they build rather complex intron arrangements. Upon closer examination of this carb intron, we found that there was a short sequence sort of in the middle, which resembled the sequence that's found in the downstream exon. We call it a pseudoexon, and you'll follow the logic of this very shortly. Now, just sort of to do our homework, typically what you do when you have these types of introns, you actually do the tedious task of trying to fold them. You want to look at the secondary structure at the RNA level, because this allows you to categorize them if they're indeed group 1 or group 2. It also allows you to categorize them into these different subcategories, group 1A, group 1B, group 1B, etc. So also this applies for group 2 introns. There's different categories. So the first module here, we call it module A, folds like a group 1, has all the parts needed to be a group 1. And it fits the categories of group 1A. The second or the last module, module C here, is also a group 1. Same thing, folds like a group 1, looks like a group 1. And then this part here, we call it the B part, which is inserted in this first one, right here in the P8 loop. It folds like a group 2. And it also has all the pieces which you would expect for a group 2. It also has identifiable these exon binding sequences, which are also required for a group 2 to splice properly, but it does not have an open reading frame. So what's the model? First of all, on paper, well, how could this splice? So basically, we developed a model just based on these different folds. And we noticed, and remember, this is where it's important to remember some of the business parts I talked about earlier on for group 1 introns. So let's just focus about group 1 introns here. So essentially, this complex intron, we have a group 1 followed by another group 1 intron module. But we have this pseudo-exon region located here. Well, when you actually fold it, it turns out that this central pseudo-exon region, at least theoretically, could serve as a sequence that can be used for the P10 interaction. And remember, this P10 interaction brings together the flanking regions of an intron, which allows the intron to be spliced out and the flanking regions to be spliced together. So if this happens, if this is used as a P10 interaction, the first group 1 intron module can splice out, but that will join the upstream exon with the second group 1 intron module. So it's like we are removing a part of the intron. Next, it turns out that this is also a suitable sequence for the P1 interaction. and still can be used for setting up a splicing configuration. And now we can use the downstream exon sequence to again set up a P10 interaction, which allows for the second intron model to splice out. This is called ratchet splicing, where you splice piece by piece using a central ratchet point as a pseudo exon, which eventually will be also removed. So eventually everything gets spliced out. Based on RNA-seq, we were able to identify the intermediates expected for the splicing pathway. So, and with regards to these types of introns, ratchet splicing was never observed in organella introns. However, ratchet splicing, sometimes called recursive splicing, has been observed in nuclear genomes when it comes to spliceosomal type introns. But remind you, spliceosomal type introns are not ribozymes. Now, what happened to the group 2 intron? Well, here we still have a bit of a question mark. Based on the RNA-seq data, we did recognize intermediates. We identified 3 circular versions of the group 2 intron, but we could not really find a definitive pathway of how it gets spliced out. We assume it gets spliced out, obviously. We did recover circular intermediates. Is it done by hydrolysis? Is it done by its EBS, IBS interactions? We don't know. This may require more work down the line. This just summarizes what ratchet splicing is, so I'm not going to go through this again in great detail. So you have 2 intron modules separated by the ratchet point. We called it a pseudoexon. In the first splicing reaction, this can be used to establish the splicing junctions, leading to the removal of intron 1. The upstream exon gets now ligated or spliced onto the intron module 2. And now we have a second round of splicing whereby the second module is removed, exons are spliced together, and the intron is gone. Now, when we're doing mitochondrial genomics, we're also looking at the DNA data as a source to maybe look at the evolution of these fungi. So I'm going to talk about this a little bit right away in more detail, but here I'm just going to point out, so we have these mitochondrial genomes. We obviously then combine them with other available genomes from other fungi in databases, including some of our own. And again, to summarize very quickly, mitochondrial DNA seems to give phylogenetic trees similar to what you would expect based on nuclear data, which is comforting to know. What we usually do, we focus on the coding regions. So we basically only use the coding regions, which is about 13, 14 core genes. We use the amino acid sequences for those. We concatenate them together, align them, and that's how we generate these trees. Just to show on the right side, we did several IP strain. Not all of them have this trintron, the one I just talked about. It's the ones I put a box next to, and there's also some which don't have this trintron. So it is an optional element, we presume, or some have it, some don't. And just to show, some members of this group have 46 introns, and other members of the genus Ophiostoma can have a lot fewer introns. So intron numbers are highly variable. During this study, Jigisha also noticed another interesting anomaly, another interesting intron I want to quickly describe without going into great detail. So this is a fungus called Ophiostoma adjuncti. Ophiostoma adjuncti has a similar life history compared to Ophiostoma ips. It's so far found only in North America. Historically, it's an interesting species. At one time, people tried to synonymize it with Ophiostoma ips, but it has sufficient physiological differences and some minor morphological differences that it's still considered to be distinct. One interesting part we found is that it has a very strange intron arrangement in the large ribosomal RNA, RNL. In the large ribosomal RNA gene, we find that it has an intron where we basically have 2 group introns separated by a mini exon. The mini exon is a single A. Now, many exons have been reported before in other organelle genomes. They're rare, and as you can imagine, they make genome annotation very challenging. Now, based on all the pieces of these group 2 introns, we can fold them, we can arrange them, and we can show that actually they can splice. So they're splicing competent. And the model we propose is that the first one here, which is at position 576. So introns are named after their nucleotide position using E. coli as a reference genome or a reference sequence. So at 576, we have a group 2, no open reading frame. It can assemble itself. It can use the upstream sequences here for their interactions, where the exon binding sequences can bind to corresponding intron binding sequences. And when it splices out, it will make available this A for the next intron. So they have to splice in sequence. This has to splice first, which then allows this A to get bumped up, so to speak. So here's my A, and my little dots moved, unfortunately. So now this A is actually the exon. It can now be used by the second intron to make these interactions to allow for it to splice out. I think it's better shown on the next slide where I simplified it. So we have one intron. It can use in the exon at the RNA level, it can interact with these sequences to remove itself. That allows this mini exon to get bumped up, so to speak. And now the second intron can use it as part of its intron binding sequence configuration to interact, as shown here, and remove itself to splice out. So one has to splice, then the next one can splice. So it's kind of a cooperative binding or cooperative splicing. You have to wait your turn. Obviously, I could drone on many different types of complex intron arrangements, but why do we find them interesting? Well, there's two reasons. One is this point, and then I'll go to the next point right away. Well, if you think about it, when you have these complex introns, which require, or intron arrangements, which require multiple steps for them to be resolved, it can potentially be a rate-limiting step. Now, it could be drift by chance, and there's no harm done, but they also could offer an opportunity for gene regulation. Now, it is known that nuclear factors are required to promote intron splicing. And so there's a potential here for what I call fine-tuning mitochondrial gene regulation in some fungi, where these nuclear mitochondrial interactions, because of these factors you need for splicing, the availability will determine if splicing occurs or at the rate of splicing. So there's a availability or possibility of what we call crosstalk between these two genomes in terms of modulating gene expression and maybe in response to all kinds of environmental cues. So that's an idea, but I think it offers certain fertile discussion. Now, I did mention sometimes we use mitochondrial genomes more for applied reasons like taxonomy. So I'm going to show just one example. One thing some of you may know about the Ophiostomatales, these fungi can be moved around. They sometimes can be exotics or introduced organisms. So it's sometimes important to distinguish your native versions from introduced species. And because morphologically they all look very similar, any morphological or molecular characters you have are definitely of benefit. So I'm going to talk specifically about Ceratocystiops, the genus. It's a group of fungi. They're usually found more in the northern hemispheres. Conifer infesting bark beetles tend to spread them around. This genus has always been defined based on producing needle-shaped or sickle-shaped or falcate-shaped ascospores and short-necked perithecia if they're present. And when they have conidial states, they are usually what's referred to as Hyala rhinocladiella-like. And I'll show a picture pretty soon. Now, here's a mitochondrial genome of Ceratocystiops minuta, which is a type. Again, there's nothing special about gene arrangement, gene numbers. We looked at 30 different strains. The sizes varied from 26 to 94 kilobases. Intron numbers varied again. Some have more, some have less. Gene numbers, synteny, highly conserved. Won't dwell on it, but you can see all of them have the same genes in the same order. Minor variations in terms of the tRNA arrangement, but very similar. What I focus right away is about the phylogenetic tree. And what we found is that species assignable to Ceratocystis actually can be separated into two clades. One, the larger one down here, is the classic one. It has the type in it, and many other species do group within the Ceratocystis. However, we found three members which form a separate clade. So again, it's highlighting to us that there's two lineages that has morphologically similar fungi in it. And maybe this second lineage, the smaller one, especially when we add more members to it, maybe should eventually become its own genus. But for now, just to summarize that little study and to give you a flavor of how we can use mitochondrial genomes to help us with the taxonomy, there seems to be two lineages which accommodate fungi which morphologically look very similar. They have short neck perithecia. Here's Minuta, the classic, the type for Ceratocystis. Here's one example of the other lineage, which may eventually represent a new genus or a separate genus. They have spores that are needle-shaped, falcate-shaped, which is very unique. Again, here's the other lineage, which could represent maybe a new genus, having the same type of ascospores here on the right. And both of them have this rather simple conidial state, which is considered a Hylarhydrocladiella-like state. Now I'm going to go through the next slides a little bit fast because I really want to just make a point. So we're finding, and it's a different group. It's not Ophiostomatales, but it's an example of, so you find a twintron or a trintron. What can you do with it? Why should you get excited about it? So this goes back a few years. Hafiz was a grad student, Mohammed, of my lab. He came across in NCBI a sequence for a mitochondrial genome, and in the RNS gene, ribosomal small subunit, he noted a really weird intron. And in this Chitomium species, Thermophilum, he noticed that in the RNS gene, there's a group intron, a large intron that's composed of a group 1 intron with an open reading frame coding for a homing endonuclease, which actually encodes, inside of it has a group 2 intron inserted. So basically, here is the group 1 intron, folds as expected, but inside of it, in the P9 region here, it has included a group 2 intron. And what's interesting is the group 2 intron interrupts that open reading frame. So theoretically, if the fungus wants to express that open reading frame for that group 1 intron, the group 2 would have to be spliced out, or you cannot make it. So we have a twintron with a group 2 intron inserted with a group 1. This just shows it again more schematically. So here's my RNS gene at that position, 1247, with reference to E. coli. We have a group 1 intron, category C2. It has an open reading frame. Inside of that reading frame is popped in this group 2 intron. Again, based on theoretical consideration, folding it on paper, so to speak, the group 2 intron actually could splice out. The flanking regions in the group 1 intron have sequences compatible with these EBS interactions. Remember, that's what a group 2 intron has to do in order to fold properly at the RNA level and splice out, bringing all the splice junctions together. And so it's all there. Now, we looked at other related fungi, which are genera, which also have that group 1 intron. They lack the group 2. So it's somewhat unique to this Chitomium. So as I said, I'll go through the next slides a bit faster. I just want to point out that the biochemistry was done. First of all, we were curious, it's good to do things on paper, but is it real? So the first thing we found out is that we could amplify this intron We could use primers that included a promoter, so we could generate RNA from it in vitro transcription. We could remove all the DNA, incubate the RNA, and see if it actually generates splicing products. And again, I have to cut this a bit short. Under various conditions, we could show that the group 2 intron can self-splice in the test tube. All we had to give it a suitable buffer and high salt, high magnesium. And we could also recover the splicing product. It spliced as predicted, which would assemble the open reading frame, allowing for that ORF theoretically to be translated. All right, so the group 2 intron works. What about the open reading frame? Once we take the group 2 intron out, is that actually a functional protein? And again, I have to cut this short a little bit. We overexpressed, we did a codon optimized version of this sequence, overexpressed it in E. coli, purified the protein, basically the homing endonuclease, and we showed that we give it the right substrate. Here's a plasmid with the substrate, an RNS gene. It will cut it. So it gets linearized. We can make PCR products based on RNS. And again, it will be cut. So the point is, the group 2 intron works and the homing endonuclease protein works. It will cleave the predicted cutting site. And again, finally, we also did some mapping exercises to show where it actually cuts specifically. And you can use a little trick. You basically have your substrate sequence, the RNS gene without the intron in it, cloned. You incubate it with the overexpressed protein. This is my HEG here. That's how we name them. Intron encoded, chitomium thermophilum, first HEG, homing endonuclease. And when it cuts, it generates a staggered cut. Now we can use a trick. T4 DNA polymerase will chew away overhangs. Then we religate that and we sequence it. We can compare the sequence before cutting and after cutting. We know what got removed, and by that we can establish an exact way of where the HEG cut. So again, we have a twintron, it has a functional group 2 and a functional homing endonuclease. So what can we do with it? Well, we built ourselves a switch. And again, proof of principle here. We designed this. So basically, we took the open reading frame for that homing endonuclease and we made two constructs, two versions. In one version, we left the original native intron, which is an A1. And in the second construct, we inserted a group 2 intron of a different category from an algae. So both of them don't have open reading frames. We inserted them in different locations. And we had to tweak a little bit to make sure that both group 2s have suitable intron binding sequences available to interact and splice out. What was the big idea? Well, we could set up a scenario where we generate an E. coli strain with two constructs, co-transformation. One plasmid here carries an antibiotic resistance marker, and it carries cloned into it an RNS gene with the target site for this HEG, as we characterized it. The other plasmid has a different antibiotic resistance marker, and it would express the homing endonuclease gene, but the group 2 intron is present. So this can only be made if the group 2 can splice out. And we have two versions of this construct. One with the original native version of the group 2, and a second construct for a separate experiment where we inserted a different category of group 2 to see if they actually work in E. coli, and if when they spliced out, it allows for functional HEG to be translated. It will cut this plasmid, remove this antibiotic resistance marker, and we can monitor cell viability based on cells losing resistance to this particular antibiotic. Now we found out, remember, these introns require high salt, high magnesium. So we have to supplement the medium with magnesium in order for optimal splicing. A little trick that can be used is you can use cobalt to antagonize magnesium uptake. So adding magnesium should turn on splicing. adding cobalt should remove magnesium and no splicing can occur. Again, very quickly, we had to prove that these constructs work in E. coli. In the presence of magnesium, both construct, left and right, work. And under high magnesium, the bands are shown in the protein is produced, which is the homing endonuclease. Both of these products were tested, and again, on the right side is just an assay to see if it cuts. So the proteins are made, and they work, they cut. And this is our switch. So we're growing in E. coli on LB, co-transformed with the two constructs. Here is for the construct with the native intron. Here is the construct with the additional, with the other group 2 intron from the algae. Now, without magnesium, just standard LB, cell viability is good. Cells are growing, cultures are growing. Add magnesium, and you can see cell viability drops. That we assume the heads are made because of splicing occurring, removing one of the antibiotic resistance marker, and our cells are starting to die off. Add cobalt, well, cells are growing just as well as on the control LB. So we got ourselves a switch. And I think that's the nice thing about these complex introns, that they're useful. We can use them to do some synthetic RNA, DNA technology. We can make switches. So what are we doing, to summarize a little bit, getting near the end? So what are we doing in my lab? Number one, we like to characterize mitochondrial genomes. And obviously, we are hunting for introns, and we're really fascinated about these more complex intron arrangements. We're also applying mitochondrial genomes to resolve taxonomic issues within this group. And as I indicated before, taxonomy in this group is complex, lots of convergent evolution, but it is important because these fungi are potentially sap-staining or pathogens, and we seem to continuously experience that these fungi get moved around globally. And so we are very concerned about biosafety in our forests, and we want to monitor what's floating around and put the correct name on these organisms. And thirdly, we're really interested in complex introns because they could be a source of interesting ribozymes and interesting enzymes that have applications in biotechnology. Now, some quick little slides. I'm just going to scan across them quickly. What applications are there? What future prospects are there? And this is not just my lab. This is in general. Why are people interested in these things? Why are people interested in homing endonucleases, introns? Well, one is homing endonucleases are site-specific DNA cutters. The target sites are usually 20 nucleotides, so they're rare cutters. One time they were that's the next big thing for genome editing, but obviously CRISPR has taken away that shine. However, some people are still working on homing endonucleases and designing them for creating specific cutting enzymes. Why are people interested in group 2 or group 1 introns? Well, you can take a group 1 intron and generate pieces for it, cut it in half, so to speak. And because of, remember, these paired interactions, they can come together in trans and still assemble a splicing competent version. So you can take part of a group 1, attach an exon, take the other part of the intron, attach another exon. The introns will come together and form in trans, setting up a splicing reaction, facilitating their removal, and allowing exons to be put together. So introns, if you work with them in trans, are actually like recombinases for RNA molecules. So there's quite a bit of interest in this technology. Group 2 introns, again, have a lot of applications. So reverse transcriptases are important as enzymes, especially nowadays that we want to do a lot of transcriptomics, and we're looking for new reverse transcriptases which are more accurate, work faster, but also group 2 introns can be engineered for genome editing. You can design them to insert into genes, but they are missing a factor, a component that can't splice out. So basically, you can use them for insertional mutagenesis. And you may have heard the word targetron. It's a commercial name that's been used, and people are still using targetron when they work with bacterial systems. And lastly, this is more new and sort of interesting. When you look at fungi and group 1 introns and group 2 introns, if you look at mammals or humans like us, we do not have ribozymes like these. We don't have group 1 introns, we don't have group 2 introns. So it has been proposed by several people that maybe in the future these could be targets for drugs, because sometimes it's very difficult to deal with human pathogens, which are fungi. So here's one proposal for group 1 introns. Here's one proposal for group 2 introns, as using them for drug targets and developing small molecules which could serve as very specific agents to deal with human pathogens. And with that, again, I would like to thank the audience. And if you have any questions, obviously, I can go back. I went through the last part a little bit fast. But I do want to acknowledge my graduate students, which obviously over the years, they're actually the ones who did the work. On the left, the individuals who worked on the mitochondrial DNA project. I identified with the asterisks those whose work I showed today. On the right, we do other projects in my lab. We work on natural products from fungi. We work on comparative genomics of fungi, finding genes associated with natural products, specifically antimicrobial compounds. So I have several students work on those. We also are interested in making materials using mushroom mycelium. And more recently, I've gotten more interested in bioinformatics, especially phylogenies based on network approaches, especially when you're dealing with situations where we have hybridization going on. And again, pictures, names are just names. Here are the pictures. And this is where I stop, just to show you the crew over the years that has been working in my lab. And I always like the one here up here. This was taken just before COVID. And the excuse I always have is we were going to go on tour, but because of COVID, we had to cancel. So we missed out. But anyways, thank you very much. And if you have any questions or if you're looking for citations that can be used, you can look on my website. And please open it up to any questions or comments.
Thanks for that, George. Very interesting systems there. Do we have any questions for the speaker? Maybe while people are formulating, I can just ask one to start. So I come away not exactly clear on the frequency with which these elements excise and so on, like what proportion of cells in a population have this happening, contemporarily, or is it a once in 10 generations this happens, or is it every single generation this is happening?
So how frequent, okay, so let's say you have one species, and within the species, how frequently would an intron move into a new location?
Yeah.
Good question. The answer is we don't know. It's not going to happen within, when we have it in the fridge, so to speak, it's not going to, if we sequence it today and we sequence it a couple of years later, we're not going to see any new introns inserted.
Mm-hmm.
When you sample across a region, so we sample different populations of the fungus, we may see that those we found in Thunder Bay may have an extra intron compared to those which we got from British Columbia. So geographically, we do see variations in the intron numbers, not like dramatic. You're looking at 1 or 2 or 3 plus or minus. But I would say within a strain, it's probably somewhat. People have done some work in yeast, but to be honest, there is no definitive number of years of how often it occurs. We do know it occurs. We do find that these are polymorphic out in nature when you just look at the introns. But the frequency, as I said, and the accurate timing of it is still somewhat undefined.
Yeah. Okay. Somebody's got a question who can't use their microphone right now, so I'll read it. I was wondering if you had tried to sequence again after successfully slicing out the antibiotic marker to see if the splicing was accurate. no insertions or off-target effects.
Yes, we did. We did. Yeah, I went through that paper really quick. So I only gave you a few highlight figures, but we did actually at every step go back, fish out the RNA or the DNA and even the protein, make sure it still works. And yes, we did confirm that the splicing occurred and the splicing occurred accurately, as predicted. Yeah.
Great. Other questions? I think Richard Hamlin maybe just joined, maybe had the time wrong, or or did I not just miss your name earlier, Richard?
That
Oh, you're muted.
I'm in Vancouver, and I thought it was as usual at 11 Eastern time
Yeah.
and 8 o'clock my time, and I just found out that it was.
Well, shucks, sorry about that.
But I may have a
Okay.
question for Richard, if you don't mind. I know I pop you in. I was going to show something here, which I know the general audience won't catch, but Dr. Hamlin might catch it, so I may as well own up to it. Ophiostomatales is a very common blue stain we come across in Canada and other parts of the world. And a few years ago, I got this frantic email from a friend of mine from Poland, Jankowiak, who's a very serious mycologist, who literally said he couldn't sleep last night because he wasn't sure what Ophiostoma ips is anymore. So it keeps some people awake. But I can sort of confer with him a little bit, because when we looked at this very carefully, now the strains we used, the isolates we used for Ophiostoma ips, they came from North America, they came from Canada, Thunder Bay area, British Columbia, Washington State, which is of course close to British Columbia, and they're all grouped together. We even have a strain in here from New Zealand, and a strain which came out of, I think, Hamlin's group work a few years ago, and a strain that came out of Wingfield's group. And, you know, that's all very legitimate names. But down here is actually a strain that is available in CBS. It was deposited in the 1940s by the original author who described Ophiostoma ips. And it is listed as the authoritative strain. The problem is a lot of the fungi in this group, and I'm sure Richard has come across this, is that type specimens frequently are missing. And type specimens in taxonomy are very important because you have to tie your concept of what you think is ips to a type. If you don't have a type, you kind of have to tie it to what other people think it is. I mean, that's the best you can do. And then you have these strains floating around. Now, it was never designated as a type. So probably the author deposited it, did not specifically say it's a type of ips. It has sometimes been used, described as a montium, which again, I don't want to get into taxonomic confusion. But so now I'm going to ask Richard, who's the real ips? What do you think? Or what are we supposed to use?
The taxonomy of these Ophiostoma is really confusing. And there's species complex, species complex, and also because they, I guess the type specimen is probably it's a culture, right?
Sorry? I think your microphone is a bit far. So yeah, the type specimen for Ophiostoma ips has been lost. The type specimen that is used by some this authoritative strain does not mean it actually is ips. It's just because it wasn't designated by the author. So there's a lot of confusion now, as you pointed out. Yeah. And that creates additional confusion along with the other complexities we come across with this group.
Yeah, yeah. No, it is, and also it it gets transmitted on wood products and on wood packing material. So it's normal that you would see them showing up in New Zealand. And then I'm suspecting that there could be also some hybridization among some closely related species. That puts in even more confusion if you just do single gene phylogeny for identification. So that I'm yeah, it's a very confusing group. Um, Unfortunately, Keith Seifert was supposed to uh, put a lot of order into this, and yeah, he was hoping that AFC would hire someone to specifically take on the Ophiostomatales, and that didn't happen. So we still we're still in a state of confusion. The Montium, though, the Montium is associated with the mountain pine beetle, but we kind of suspect that it's not it's not closely associated with the mountain pine beetle as the Grossmania and the Leptographiums are. But so we think it could be Montium could be more of a generalist. So maybe that's why it shows up. You said it shows up here. I can't see it. Oh
Well,
yeah,
it does
there.
show up
Yep,
here,
yep.
but I mean, it's outside of the clade that what we think are what we think
Ips.
in general is IPS.
Yeah.
It's close to adjuncti, which again morphologically is not surprising. Morphologically, they're all very similar. As I said, this IPS is a question mark, although some people list it as authoritative. It may have to be re-examined. Actually, the one below here was at one time also called IPS. It comes from New Zealand too, but there are some morphological differences here. So that one we could easily explain away, so to speak. And this is just historical issues of confusion of type. And these are the ones, as I said, it's not just, these include not just from us, but from other groups. So they all seem to grow together. There's a tree based on Bayesian, and the node support is quite here, 90%. So, I mean, so that does probably mean these are all the IPS as we normally recognize them here. Yeah, just to point that out, yeah, the taxonomy definitely still needs a lot of work.
It's a mess. Yeah. It needs a dedicated person to dive into it. And now also with whole genome, like I said, I think there's some hybridization. We saw it in Ophiostoma novoulmi and ulmi, and so I think there's some hybridizations there that could really confuse the taxonomy. Oh, I'm so sad I missed your talk. I was looking forward to it. I had it in my agenda for 8 o'clock Vancouver time, and I was just logging in. So.
Well, I think they did record it. If you want to go
Oh
to the
yes,
recording of it.
perfect. Okay, I will listen to it on the recording. Yeah.
So we are at that time when we should transition here. But yeah, George, thanks a lot. Enjoyed
Thanks,
that.
George, and
Okay.
see you soon. Bye.
Yeah.
Thank you, everyone. So fellows that are interested, you're welcome to stay, and we'll take a brief pause for Dr. Hausner for like, I don't know, 3 minutes, or if you don't need a pause, we can go right away. It's up to you.
I'm fine.
Okay, so thank you, everyone. Okay, we don't have a lot of fellows that are there, but Christine, Mathieu, or I don't remember
Harinita.
Okay, so first name, sorry. If you wanna take over for the Q&A, Mathieu, do you wanna do it, or Christine?
Maybe I can do it.
Yeah, do it, please. Go ahead.
Yeah, sure.
Since I don't see you, I don't know if it's normal or we don't see you.
I'm in the biker lab.
Oh, I see.
there,
Okay.
But I'm just in a different committee, not in the seminar Q&A committee. So yeah, it's okay.
Okay.
But,
Well, go ahead.
hello, Dr. Georg Hausner. Thank you for the wonderful presentation. So now, if any of the fellows have question, they can go ahead. Or maybe I can ask a question. So it was a great talk. Personally, in my undergraduate, I had an opportunity to work with beetles from mountain pine beetle, spruce bark beetle, western bark beetle. So I was in the lab in University of Northern British Columbia under Professor Dr. Kalindi Morgan. And we were trying to explore some of the important nitrogen-containing secondary metabolites. So we were looking at the polyketides thing in different microorganisms, not only fungi, bacteria, actinomycetes, and different fungi that are present in the microbiome of these beetle that are present in the different types of trees. So we did we collected beetle sample from these mountains in different location in British Columbia. And then we isolated the microorganism in them, and we did a basic analysis of genome analysis and biosynthetic gene cluster. And since it's like a preliminary kind of like a study, we were able to see this interesting polyketide and interesting secondary metabolite that present in these. So now looking at your research, looking at the different mechanism in their transcription process, and especially this interesting exon group 2 and intron group 1, group 2 mechanism that's especially specific present in these fungi. It was really interesting to know. And I also did see that your group also doing some secondary metabolites. So are you guys interested in any class of secondary metabolites, or did you guys discover anything that present in these pathogen, something like that? Yeah.
So yeah, to go back, so yes, actually we are looking at a comparative approach. So actually 2 of my students are working on secondary metabolites, one specifically on the Ophiostomatales. So that's awesome. And she has sequenced quite a few genomes. And of course, like everybody else, we go with anti-smash, we fish out gene clusters that are identified. Are we looking for something specific at this point? Now, in her study, it's a survey. So she's been looking at different members of the Ophiostomatales with different lifestyles to see if there's a difference in the biosynthetic gene cluster. So her work is more the big picture. We're also looking at the Cas enzyme distribution and the typical genome annotation, COG, gene clusters and categories. So with regards to secondary metabolites and secondary biosynthetic gene clusters, to be honest, what we found is quite, I have to be careful to say conserved, but they seem to have very similar clusters. And as I said, we're looking across all 5 categories of secondary metabolites. So we're not just focused on the polyketide, but of course they are very dominant, along with the non-ribosomal types and the hybrid types. But it seems to be that within Leptographium, so we're comparing the different genera. The pathogenic members sometimes have more. That could be more by chance, by drift. So we're getting into lifestyle observations, the generalists versus the specialists. But we have not found, in case anything potentially highly useful as an antimicrobial, but we have found categories, and we're trying to eventually establish combinations and see how it links to lifestyle. So this is work in progress. That doesn't quite answer your question, I guess. But I think there's a lot of interest in secondary metabolites and biosynthetic gene clusters, partially because there could be interesting compounds, but partially also there could be compounds which are needed as signals, like terpenes, to attract their beetle associates, etc. There may also be compounds hidden in there which might be important in terms of host pathogen interactions, etc. But I think at this stage, we are just keeping it very broad.
Thank you for that. And I think we have a question from Christine. So the question is, I'm quite interested to hear more about how far you work with the genome editing design is going now. Have you tried to utilize this method for something else or other biotech applications?
Genome editing. So I guess, sorry, there was 2 parts, or is it mostly about genome editing, or is it also genome annotation?
It's like a genome editing design and how you
editing.
utilize
Sorry,
this method for any biotech application.
Yeah.
So yeah, it's a question. Yeah.
No, I misheard. Sorry, that's okay. At this point, the answer is no. I mean, we are still finding, we have patented a few homing endonucleases. There's always the easy part. The easy part, I will admit, is finding them, is doing the basic characterization that they work. The hard part is to then get the real money. And this is now where it gets into the cruel world of funds and getting grants and finding potentially industrial partners. So we have reached out, and now homing endonucleases, I will have to admit, they're a hard sell now. With CRISPR-Cas, which is much more affordable, already established, so you can't compete with CRISPR. On the RNA, while I think it's still very much in the lab where people are doing these trans-splicing, it's a nice model, it has tremendous potential, but I don't think we're there yet to get anybody interested to give us a lot more money. So to be totally honest, it's at the level of basic science, but we haven't had yet the opportunity to go beyond it. That doesn't mean we're not looking, but I think the challenge in science is you have an idea in the lab and you can do some really great science and publish it, but the next big hurdle is to actually sell it if you want to go big, so to speak, because then it gets very costly. So the answer is we have not proceeded beyond basic science, which is the characterization and just trying to understand how these parts work. And that's where we're at.
Then there is also a follow-up question from Kristen. I was wondering if you're familiar with integrons, which are also mobile elements. So
Yeah.
probably referring.
Yeah, I mean, when you look at bacteria, they have all kinds of mobile elements, integrons and insertion elements and transposons. And what makes, I guess, our mobile introns unique in a way is that they are more specific in terms of, I always say they're mimics. And what I mean with that is group 1 introns, group 1 and group 2 introns are mobile elements, but they pretend to be introns. They mimic to be introns. So they will sit inside a gene. They cannot sit outside of genes. They benefit by being in a gene by taking advantage of all the regulatory pieces a gene offers, like a promoter. So that allows them to be transcribed. If these introns encode proteins, they can be translated. So they basically, you call them like little freeloaders, hitchhikers. Introns, integrons, transposon insertion sites, integrons, insertion elements, and transposons, sorry. They can be in intergenic regions. They can sometimes by accident disrupt a gene and cause a mutation that way. Whereas the mobile introns, that's their lifestyle. They need to live inside genes. But definitely the world of mobile elements is quite large. It's quite fascinating.
Thank you, Dr. Hausner. And does anyone have any questions to follow up with Dr. Georg Hausner?
Yeah, I would have a question. It's less about the science, more about like your the academic background and stuff. Like, how did you first hear about mitochondria and mobile genetic elements and stuff like that? Was this something that inspired you, or where did you start the research, and then how did you end up studying what you're studying right now?
Yeah, this is always a proverbial fair question. So I started out as, I go way back. As an undergraduate, don't ask me why, I took a mycology course and I really got hooked on fungi. So number one, so that was it. Once I had fungi in my blood, that was it. Then I did a master's, which was more fungal physiology. And I still enjoyed fungi, but we're now talking and giving away my age. We're in the around 85. And that's sort of just when DNA became big. I know this sounds very ancient, but And I realized if I'm going to do something next, and I also was starting to think career-wise, I got to add some more modern technologies into my tool belt, so to speak. So a project came up to work on the taxonomy of the Ophiostomatales, and to be honest, at that point, I never heard of the Ophiostomatales, using molecular markers, using DNA sequences. And I know this is in the 80s where this was really hot. You know, people still worked on going back to Carl Rose and the 16S, and everybody was doing 16S. So I thought, wow, on fungi, I'm in. So that started my PhD. My PhD was mostly working on the taxonomy of the Ophiostomatales. That's how I got to know them. Got to learn how to sequence the old school by Sanger, by hand, running gels, doing X-rays, reading. Anyways, old school. And so I did that, and it all worked out great. We got a lot of work done. But towards the end of my PhD, totally by chance, at a conference, I ran into somebody called Helmut Bertrand, who was working on Neurospora crassa, and he wanted to recruit somebody who knows more about fungi, has a broader background, and knows how to sequence. At that time, that was a skill good enough to get a job. So, all right. So I did a postdoc at Michigan State, working on Neurospora crassa, and yeah, their model was mitochondrial DNA. And the reason I went there, because I wanted to learn more real molecular biology, cloning, sequencing, making mutants. And it wasn't necessarily that I was into mitochondria. I was just more like fungi, and I can learn more techniques. And so that's where I learned about cloning and doing RNA work and doing splicing work. And I also got into mitochondria. And I thought that kind of cool. And meanwhile, I had a side project working on Cryphonectra parasitica, which some of you may know is a chestnut blight fungus. And they were working with hypervirulent strains. They were working with strains which came from northern Michigan, which didn't kill the tree, would infect. And they had mitochondrial defects. And that got me curious. Now, number one, we found one strain and a couple of other strains where the defect could actually be eventually linked to a intron. And an intron that seems to be splicing poorly. And that that shows you what happens when things go, I wouldn't say wrong. Being hypervirulent actually has advantages, because if a pathogen kills all its food, it's got nothing to eat. So sometimes populations become hypervirulent because they're running out of hosts, and it's actually beneficial to not go for the kill, but just be a more silent passenger. Anyway, that got me into introns. And then we sequenced it, and that was the old school, folding it by hand. And I thought, oh, these things are kind of cool. And then I found another intron in the bug, not related to pathogenicity. And that kind of got me going into intron. We're talking early 90s, when there wasn't that much work yet done on them. So I remember when I wrote the first paper on introns, I only had 30 citations because there was hardly any work available to cite, except the classics. Now that's about as many papers that get published a week. I mean, it's just tremendous. But anyway, that got me into introns. And ironically, after Michigan State, when I came back to Canada, there was no jobs anywhere, although I did find a job as a research scientist in Agriculture Canada. But there I worked on something completely different. They were looking for somebody who can do PCR and sequencing and develop molecular markers to find resistance genes in different crops. So for a couple of years, that's what I worked on. Eventually, an opportunity opened up in Calgary where they worked on group 2 introns. And that got me back in the game, so to speak, to go back to group 2 introns and then group 1 introns. And so it's kind of a little bit a zigzag pathway here. Sometimes it's just a matter of opportunity. Sometimes it's a matter of looking at opportunities to learn new skills. And it just happened to be in a lab that works on mitochondria. Within that lab, I discovered introns. That got me hooked. I had to leave introns behind for a while. Then I came back to it because an opportunity arose. And when I set up my lab in Manitoba, which is sort of a circle, I did my PhD at U of M. I come back eventually to U of M. The culture collection was still here. which I worked on in my PhD. So it's kind of an opportunity to think, wait a minute, I got all these Ophiostomatales still here. So I might as well go back to that collection and start working on them from a point of view of taxonomy again, because I know what I'm doing. But also I can start working on, nobody has looked at at that time, the mitochondrial genomes of these fungi, and start exploring also for introns. So that's kind of the journey here. It wasn't a straight line.
Nice. Thank you very much for sharing.
Okay, I think that's the most of the question from the fellows. Thank you, Dr. Georg Hausner, for your wonderful presentation.
Okay, well, thank
Thank
you
you.
very much. So just to throw a question at the audience before I leave or before we shut it. I mean, you can ask me anything about my history and my work. So, I mean, I know you're a more plant pathology, evolution type background. So I have to admit, I was thinking about my talk. It's like, meh. Of course, the Ophiostomatales are a classic group that should get